Good afternoon, everybody. I wanted to thank you for joining the Bioquell's presentation today for new methods for accelerating stem cell growth. We're pleased to have with us today Dr. Melissa Reynolds. Dr. Reynolds is an assistant professor of chemistry with Colorado State University. She is also the founder of the biotechnology company Diazimed Incorporated. Dr. Reynolds and her research team have been nationally recognized for their work with materials that accelerate wound healing. In the past, she was named Educator of the Year by the Colorado Bioscience Association in 2011, and she was selected as one of the 6 inaugural Bodicher investigators in 2010. We look forward to having her share some of her experience and knowledge with us today in this webinar. To everyone viewing this webinar, at the end of the presentation, there'll be a 10-minute session for questions. Please feel free to send us your questions to the right-hand side of the screen, and if we can't answer them in this session, we will get back to you after the presentation has concluded. With all that said, I'd like to turn it over to Dr. Reynolds. Dr. Reynolds, please go ahead with the presentation.
All right, thank you for that wonderful introduction, and for everyone that's taking the time today to listen to this webinar. I would be remiss to not recognize first the folks that have been involved in this work, which is my graduate students at Colorado State University. So today what I wanted to talk to you about in relationship to cell growth and cell methods and thinking about how we can control toxicity and the behaviors of cells, I'd like to put you into my world for a couple minutes, and that is the notion of thinking about how medical device technologies interface with cells. And maybe a better way to think about this really is more of how do cells respond to technologies that are needed to either improve the quality of life of patients or necessary treatments for being able to treat an infection or some type of illness. So we are all familiar with medical devices in different forms or fashion. Everything from contact lenses to implantable devices to glucose test strips. Most of us have heard about stents, which are devices that will open up arteries. There's a variety of other anti-infective type of devices as well. So because we're all familiar with this, I think sometimes we take for granted really that interface between the materials that these devices are made out of, the risk to patients, and then how also these devices interface with cells. And as most of you are aware, some of the newest technologies moving forward are thinking about cell-based therapies. And so to me, that's how we really connect all of these different pieces. The notion that we do want to think about better interventions for patient care, but we have to think about both the synthetic materials, the cellular materials, and how they interact. So when we think about whether it be contact lenses or other types of maybe not permanently implanted devices, daily cleaning is oftentimes needed to decrease the risk of infection. The issue, though, however, with implanted devices is they cannot be removed for daily cleaning. So we need the cellular response to these devices to be very, very controlled. Some common examples of devices that some of you may be familiar with are devices such as stents, which is the picture on the lower left, metal mesh that's used to expand a blood vessel, vascular grafts, which are artificial blood vessels. It could be used especially in chronic dialysis patients or, again, in cases of heart attack. Other types of hernia meshes or tissue repair devices where you need tissues and cells to interact with the mesh to accept that into the body. In all of these cases, a lot of times there's compromises and there's failure of the devices due to infection as well as thrombosis. The idea being is that we aren't able to control the cellular response. We aren't able to understand how those cells are interacting with materials in a way to make improved types of technologies. So the question that I oftentimes like to think about is, how can cell studies really guide our development processes for improved therapeutics? I will argue that one of the major issues that we have as a community is that we need to better think about our characterization strategies when developing these new types of therapeutics. We need to think not only about what might be happening at the material or at the drug interface, but also what might be happening at the surface when cells with proteins interact with our therapeutics. And we also need to think about the biology. So if we're going to create more advanced therapeutics, whether they be cell-based or materials-based, we have to understand not only the interface, but we have to understand how the cells are acting on the material, and conversely, how the material is acting on the cells. So I'll argue with you today that we probably need to think about this more comprehensively in maybe a four-pronged approach. The idea being, one, to think about bulk material, so drug loading, morphology, thermal stability, which of course is important for manufacturing. We need to think about surface components, whether that be wettability or topography. We need to think about what might be happening in the solution, so release rates of therapeutics, growth rates of cells, degradation of materials, whether that be ideal or not ideal, and then the biology. What is the mode of action that we want the intervention to have on the cells, and what type of effect we want the cells to have on the intervention? And of course, underlying all of this is the modality that most of the time we're looking at trying to also prevent toxicity. So the idea being is that in many types of therapeutics, we want a therapeutic mode of action to hit a certain biochemical cycle that we want to have an influence on without any additional toxicity. And when we think about all of these together, it's quite a challenge to be able to consider all of these at one time to think about more advanced characterization strategies. In my world, we think a lot about wound healing devices and materials. We also then think about regenerative materials towards being able to have improved performance. So one of the major considerations that we have is really tissue disruption as well as clotting. So if you have to put a material into a physiological system, on the right-hand side, I'm showing you all of the different complications that typically occur. And depending on placement of these types of devices, whether they're cellular-based or whether or not they're synthetic materials-based, one complication may have a greater impact than another. And in the desired world, we really want longer-lasting therapies that are safer, more effective, and ideally less expensive, and most importantly, with less side effects. As I talked about in the previous slide, there's always toxicity concerns. There's other unintended consequences that we oftentimes can't necessarily think about when we're doing the initial design that come up that we have to address at some point. And if we really think about longer-lasting materials or longer-lasting therapies to treat the problems that I've identified on the right-hand side, what we're really saying is that we want to prevent the interactions that lead to failure or rejection of the device or of the therapy, and we want to think more about how we can do this in a fashion that's not going to disrupt necessary biochemical processes. Obviously, when we have therapeutic treatments, we need to either upregulate or downregulate a particular process in order to have the effect that we're looking for. But at the same time, we don't want to be causing effects in other types of processes that are ongoing. This is one of the major problems with a lot of current therapies and treatments. We end up solving one problem and creating another problem simultaneously. The movement to cell-based therapies, I think, is a great movement in thinking about being able to integrate within a biological system rather than creating a microsystem within biology that could create additional problems. So I think about this by asking really two questions. One, can we think about chemistries? Can we think about processes? Can we think about cell modalities that allow us to direct proteins and other cells locally where we want the sphere of influence to exist? An example that I love to give is the notion that if I have a cut on my arm, I don't need my big toe to feel the effect of trying to stop the bleeding in my arm. And so what we're really thinking about in the newest types of therapies, especially cellular therapies, is can we actually direct really locally where we want the action to occur? And I will argue that the second thing that we need to think about for better treatments is to understand the biology-material interface, and that's including the cellular behavior. What allows cells to be able to grow in specific regions unimpeded? What has actually been controls their ability to not function when needed? And are there certain triggers that we could take advantage of to make better cell-based therapies? I also like to think about this in terms of biological impact. So the idea of translating from in vitro environments to in vivo environments that include human patients. And that is, a lot of times when we need to use our therapies, we have to think about two different influences towards the use of the therapy. One being the surface of whatever that device is or that therapy that we're using. And then secondarily, especially for medical device technologies, the notion that injury is an important process that occurs when physicians are placing these devices. So if we think about either diabetic patients or cardiovascular patients that require stents, in all of these cases, a physician needs to basically perform an operation in order to use these technologies. So we have to then think about the therapies in terms of treating both the surface tissue-cell interactions as well as the injury that's just occurred. Because injury is a necessary side effect of medical device treatment, if we're developing new therapies, we need to consider both. One of the major cells that are destroyed in a lot of cardiovascular treatments are endothelial cells. There's biochemical pathways that produce nitric oxide that are usually cease to exist. There's also surface chemistry on the outside of cells that send signaling agents that also tend to be disrupted in the placement of these types of devices. So when we think about cellular components and cellular therapies, the biochemical messaging systems required to maintain cell function are disrupted. So when we try to move then from in vitro cell studies to actually in vivo cell behavior, the question starts to become, how can you replicate or create this function in living systems? So my group thinks a lot about how we can make either materials or cell motifs in ways that we can be mediators of biological responses in opposition to thinking about shutting off biological responses. So in the cases of injuries, a lot of times nitric oxide is not produced at the surface, and if we can try to put that function back, we can prevent infection from occurring at the surface. We can promote angiogenesis, and we can reproliferate fibroblasts in that region until cells are reproduced at the level or regenerated to produce that function on their own. The second piece then would be device integration. Again, if you can actually get the cells to integrate around the device in a healthy way until they return to their function, then you actually have a device or material interface that can promote additional cellular functions as well. And this is analogous also to cell-based therapies, where if you can actually get cells to proliferate appropriately, and I want to highlight the word appropriate here, in a way that then has function that's in a normal biochemical process to uninjured cells, what could happen around that device then is regeneration and incorporation instead of rejection. So there's a variety of different mediators biologically that one can use to test some of these ideas. My group focuses on using nitric oxide as one potential mediator of these biological processes, most noticeably because the compound has a very relatively short half-life compared to a lot of other types of drug therapies. And the reason why this becomes important to us is because we focus a lot on permanently implanted materials, and having a drug being released from the material and going into the circulation doesn't keep the effect of the drug localized. If we use a very short half-life drug, and I mean on the order of milliseconds, not on the order of seconds, hours, and so forth, then what you get is localized action at the surface of the material or at the surfaces of the cells that are very localized to a potential region instead of more of a systemic effect. The challenge with this, of course, is being able to store and deliver on command over the time frame that's needed for any particular application. The other important recognition here is that nitric oxide has, as with all drugs, a variety of different actions, both cell protective as well as cell death. And so trying to find that balance locally becomes very important when thinking about these types of therapies. So if you want to use a cell-based therapy that is going to be able to reproduce endothelial cells by actually then promoting cells around by sending in signaling agents, you want to make sure that you're going to be able to provide the right dosage and an effect that's going to recruit those cells rather than kill the cells around the site of the intervention. So I want to tell you a couple short stories today about how we go about thinking about directing different cell behaviors and promoting growth in a way that's not toxic. So the first short story that I want to tell you today is around the question of how can we direct cell behavior. So we think about cells not only from a tissue engineering standpoint, but also in terms of clotting and blood vessel cells. So in normal cases of clotting in a natural vessel, platelets will adhere, platelets will activate, cells adhere. The vessel is repaired under normal circumstances, blocked under extreme circumstances. And this is considered a good thing when the vessel is repaired because then you'll stop bleeding. Okay? In the case of a synthetic implant, the process is slightly different in the fact that proteins absorb, platelets and cells adhere and activate. More cells adhere to the surface, more proteins adhere. Usually this leads to a blockage in the blood vessel, cells that are upregulated in a negative way to actually then send back chemical signaling messages to cells to recruit other cells, and you end up either having a clot or some type of blockage that occurs. Current anti-clotting strategies think about trying to control these cells by giving, again, a systemic treatment option that either knocks out the platelets or the cells from being able to activate when needed, or by being able to create more of a systemic effect, which causes potential risk for bleeding. So we think about controlling cells in this case in terms of getting from systemic therapies to surface localized therapies. When we think about this, though, there's a consideration, and I started this presentation by saying that we need to think about the whole system. So in terms of the materials properties, what is the actual influence on the blood cells or other types of cells that might lead us to having the therapeutic action that we so desire? So in a lot of cases with cell-based therapies, there's either some type of substrate or there's some type of interaction that you need to occur in order to create a growth medium that these cells can actually proliferate on appropriately. So there's certain materials features then that become important, and examples of these would be like wettability, roughness, composition, the actual time kinetic profiles of the drug release. By being able to measure all of these different components, we can better isolate the contribution for the actual drug component that is needed to actually either promote or prevent cellular behavior that we so desire. So we have actually done quite a few measurements to try to isolate and look at what are the relative effects of, again, our monocompound nitric oxide on being able to direct or control cellular behavior. And so we began to look at this from a two-phase approach, one from protein surface interactions and the other from cell surface interactions. We use protein absorption methods to be able to look at the relative binding of different proteins to our surfaces. We do this both to look, again, like I said, at proteins as well as platelets, and then as I finish or go through this talk, we'll also talk a little bit about recruiting cells to these scaffolds as well. Well, we ended up discovering a very interesting result, and that is with our surfaces, we're actually able to recruit a very high blood protein to our surfaces, known as fibrinogen. Most folks that would look at this data would say that this is counterintuitive for looking at clot formation. One would say normally that the least amount of fibrinogen is your best surface for being, quote, biocompatible. We actually found that the presence of nitric oxide in a dose-dependent manner will actually turn on additional fibrinogen to bind to our surfaces, which is very different than the other types of proteins in the blood as well. What we found, though, is that this fibrinogen is then no longer active to participate in the clotting cascade. In other words, platelets will not bind to the fibrinogen and therefore not becoming activated, therefore rendering a surface that then will not clot. And we know that we've isolated the effect of nitric oxide rather than the other materials parameters based on our extensive sample preparation method as well as characterization on the surface. We then went on to look at the relative effects of flow because, again, in a biological system, blood doesn't just sit in a petri dish. It continues to flow through our veins and arteries. In collaboration with Keith Mead's group at Colorado School of Mines, we actually found that our surfaces, when producing enough nitric oxide, will prevent, again, platelet aggregation under adhesion and under shear flow conditions. And if you look at the graph on the upper right, what you'll see is that we can actually now very well correlate the relative platelet aggregation number to the dosages of nitric oxide that we get off of these microfluidic devices. This allows us to tune in, especially for cardiovascular applications, the therapeutic amount that we can actually get to promote different types of cellular behavior. I will note that through a variety of different cytotoxicity tests that we have done on endothelial cells, these levels of nitric oxide do not cause decreased performance or cell death of the endothelial cells within that region as well. So again, bringing back the function of one type of cell and making sure that we don't knock out the function of the other, we've been able to demonstrate through a series of different types of cell studies. We've also done some platelet assay testing at the same time. One of the concerns that oftentimes arises in these types of methodologies, especially with cell assays, is whether or not the cells are alive or dead when you're observing whether or not they adhere to surfaces. Oftentimes they will not adhere because they are not functional or alive. And so we've done a series of tests to make sure that all of the cells that we go through and we evaluate on our surfaces or on our either water-soluble materials tend to have activity after the function is over. So we actually have a platelet assay test where we can actually then pool plasma, look at relative fibrinogen absorption, platelet adhesion, as well as cell viability in a microtiter plate assay, and compare that both to our control as well as our active surfaces, and can demonstrate then that in our case that all of the platelets remain at least reversibly active, even if they're knocked out for a short period of time at the interface of the material. So what we actually find in this case is that we can actually demonstrate, and this is looking at G and H, which is our active surfaces, that we get significantly less platelets onto surfaces that release nitric oxide, and at the same time we see functional platelets that are actually there available for clotting when needed. So we've been able to look at both cellular behavior of the platelets or cells of interest as well as their dose response profile, and also checking to make sure that the cells are functional. So my second short story goes on to this notion of, is the observed behavior due to the intervention or the experimental design? So the studies that I just presented to you on some of the platelet work, and I alluded to some of our endothelial cell work, really looks at whether or not we see a difference in cellular behavior at an interface or at a surface, with the idea being is that if we can understand that, we might be able to lead to longer-term tissue engineering types of methods or designs. The question then becomes, are we doing the right experiments? And are the experiments and how we have them set up appropriate for answering the question about cellular behavior? So I usually think about this from a perspective of not necessarily what is already being done, but what is it that I really want to look at? So the idea being here, what is the sample? What is the sample? What is the information from the sample that I want to know? What piece am I actually measuring? And how does that relate to what data is collected? Is the data really related to what I want to know? And is the method appropriate for the question that I'm being asked? So if I go back and I think about the platelet studies that we ran, I would think about the question of, well, are the platelets adhering to the surface? The answer may be no, and that may be the result that I might expect or desire. But if the platelets aren't functional, then I'm not really measuring the effect of my surface because the platelets aren't activated enough to be able to stick even in an agonist situation. So the second piece of that is that if I want to know if my nitric oxide or my drug is having an effect, how do I eliminate all the carrier or scaffold component out of that to make sure that I understand the influence truly on the cells and not other factors that are being involved. So if I want to know about a material, or I want to know about the material on the cells, I want to think about how I can end up measuring that feature. And then that secondarily leads me into the notion of, does the way that I perform the experiment give accurate information regarding what I want to know? So we spent actually quite a bit of time in my group trying to make sure that we have accurate measurements for thinking about these questions when aligning to different types of cells. So one of the considerations that we think about with methodology considerations, especially related to cells, is do the nutrients and media sustain cell growth? Is the drug dosage equal to the drug that's available to cells? Are the appropriate positive, negative, and reagent controls used? Is the time frame of the analytical measurement method appropriate for the intended use? And are the observation times relevant? And that kind of links us into, I think, the translational piece here, and that is all of this information then clinically relevant to report, or allows us to go from the in vitro to the in vivo system? So I want to tell you a quick little short story about what turns out not to be true. So one of the, I think, myths that goes on a lot of times is this notion that the drug that you're giving into either phosphate buffer or into cell media actually equals the available drug that's available to the cells in the solution that you're looking at. And what I will argue is that this isn't always the case. It's especially not the case for highly reactive types of species. So one of the studies that we have done recently and shown is that if you have a carrier for nitric oxide and you put it into a solution of cell media, you do not have all of the amount of nitric oxide available to cells that would be calculated if you based it just on the amount of nitric oxide donor or drug that you were getting. If you look at the graph on the right-hand side, what you're going to see is you see a series of different types of NO donors, which the identity of them is not as important, except that when you look at the different types of environments that they're in, you'll see a drop in the amount of nitric oxide that becomes available. So, of course, in phosphate buffer, that's the dark black line. You actually see that we have about what we would expect based on their half-life. Again, in endothelial cell media, again, it's very similar. But when we move to neurobasal media, even though we have the same concentration of drug, we actually have a much less amount of nitric oxide that becomes available to cells. So this gets us back to thinking about, well, how much drug or how much nitric oxide do you need to have an effect on cells? Well, if we would have used old or more traditional methodology, we would actually report much higher values. It turns out that those higher values tend to be toxic to other types of cells, so therefore they wouldn't be able to move us very far forward in thinking about types of devices. If we go back then and we look at data like I'm showing you here, what we end up finding is that the amount of nitric oxide that we produce, we can start thinking about how we can translate that then to devices. Well, we ended up finding out that there's a tryptophan ribofran complex that gets formed in media. This is actually a scavenging agent that's available for nitric oxide. So the bioavailable NO to the cell to be able to control or direct them is dependent then on the types of media that we tend to use in cell studies. So once we have this information, we actually then could work with a collaborator, Dr. Vi at Colorado State University, who's actually interested in blindness. And one of the questions that he had was, can nitric oxide be improved or used to improve vision of low light? And if so, how much is needed? So again, now that we have this greater understanding of how much nitric oxide might be available to cells, we were able to work with him to look at goldfish retina cells to determine the amount of nitric oxide needed to actually improve vision or improve performance of these cells under low light conditions. And we found, again, a nitric oxide dosage that if you look at the right-hand side, kind of bottom column, you'll see is substantially different than if we hadn't taken this into account in our measurement system. The result of these experiments was that stimulus by NO did improve the low light detection, but at a much smaller dosage that was previously reported or thought possible. So dosage-dependent activity of nitric oxide, or I would say any other therapeutic, is important, and it requires accurate measurement of the drug that's available for use or for binding to different types of cells. So this leads us into, again, another materials design consideration, and that is, are we actually thinking about the contributors of cell viability? So we worked with a variety of different cells, fibroblasts, endothelial cells. We have done a little bit of stem cell work, osteoblasts, and keratinocytes. We've thought about making our active materials both in dissolved form as well as plated. We tend to synthesize most all of our materials, so we run a lot of reagent controls to make sure it's not the material itself that's having an effect, but our drug of choice. We also look at cells alone as our method control and then use a toxic polymer to be our negative control, again, to test the validity of our method. what we end up finding in these experiments is really the identification of the contributions to cell behavior, both the protective and toxic effects of the derivatizations that we make in each of our materials. And I think sometimes, especially for more synthetic derived materials, a lot of times we'll skip some of the reagent control steps along the way, and I think we have to take a step back and also take into consideration the influence of those on the cells as well, because it's really the collective group that's going to be able to contribute overall to the cell viability that we observe. So two kind of quicker examples of how we've actually been able to practice this. One is with biodegradable materials in medicine, hernia patches, tissue engineering devices. We need usually extracellular matrix change components, vascular smooth muscle cells. We have to think about endothelial cell proliferation. We also have to think about platelet aggregation, inflammation, as well as infection. And so what we have done is we have created different types of biodegradable materials and looked to see how each of these different cell types end up responding to these materials. We also think that these might be great material scaffolds for being able to create cellular-based device, because they can recruit the right appropriate cells in the right orientation and then dissolve away after the cells create their own environment. So we have worked with a range of different materials, everything from enzymatically degradable materials to hydrolytic materials. We derivatize them in different ways to put different types of nitric oxide or other mediators on the surfaces, and then we process them in a way that's going to, we hope, allow us to recruit appropriate cells. Without going too much into the characterization of all of these materials, I do want to point out that we have done a lot of work to look at any byproduct formation as well as other types of interferences that might contribute to cell behavior. Nitric oxide, as well as many other drugs, have a variety of different interactions with cells, proteins, other types of biological molecules that could give rise to different types of cellular behaviors that we've looked at. So we've actually spent a lot of time correlating the behavior of materials and their chemistries to the different types of cellular behaviors that we use. And I think, again, this is very important for thinking about cellular-based therapies, because small changes in the chemistry or the interactions could have huge influences on biological processes. We then move and fabricate these materials into forms that actually could be useful. We have tried a lot of times to make nanofibers out of these materials to make mats the cells will be able to have the right porosity to bind to. And we have been able to do this. The materials maintain their nitric oxide release capabilities, and we can get some fine nanostructure properties that mimic the extracellular matrix. These materials also tend to remain their structures after, at least initially, in aqueous conditions. In other words, there's a structure in place long enough for cells to actually deposit onto the surface. So we've actually been able to take into account the bulk, surface, and biological effects of these materials. We get very high kill efficiencies of Obama E. coli as well as Staph aureus. We see a decrease in platelet activation. We get cytotoxicity scores of 0 using the ISO standards. We do see endothelial cell growth on these surfaces, and cell inflammation has been decreased. My final very quick story is going to be a new class of materials that really hasn't been used in biological applications before, and that's a class of metal organic frameworks that could be used as catalysts to support therapeutics at a surface. Again, we have gone through the analytical chemistry to prepare methods to be able to monitor the production of nitric oxide in real time. And what I'm showing you here is actually a control experiment where we aren't measuring any nitric oxide, again, to demonstrate that unless we perturb the system, we won't be able to produce nitric oxide. We can then take these crystals and we can demonstrate catalytic turnover time and time again. The picture on the left is basically taking out the crystals, brushing them off, putting them back in, and looking for the reaction to occur. The kinetics of this does not change over time, so we get prolonged potential release of nitric oxide at the surfaces of these devices. These materials remain structurally stable, which is important because thinking about their impact on cells, if we degrade any of the materials, then they have the potential to either cause toxicity effects. The materials on the left show that even under cell culture conditions in blood and so forth, they remain intact, so we don't leach any toxic metals out, and their catalytic function remains similar both before and after. So the material is functional even after exposure to cells, and the cells do not seem to respond negatively to the material. We then put these into different medical device platforms. Extruded tubing is what I'm showing you on the left, grown off of the surface through a simple manufacturing washing process is on the right. And these materials are highly functional without showing any leaching or toxicity to cells using a variety of ISO standard models. So what I hope that I've kind of given you some examples of today are really thinking about biomaterials in the context of cellular behavior as well as controlling cellular growth. There's two different platforms that my group has worked a lot with, and that is these metal-organic framework catalysts as well as polymers for biomaterials. And again, these materials show very nice safety profiles. They seem to have very good control of different types of cellular behavior, and their material properties tend to match the applications that we've been designing for. As a kind of a wrap-up, I just want to remind everyone that I think methods, materials, and cells methods must match. So the bulk, surface, solution, and the biology, we need to understand all of those if it's going to lead us to creating better cellular therapies. So if we think about an in-depth look at the efficacy and toxicity of biomaterials, I would say we have to think about how do we direct cell behavior? Are the methodologies appropriate for the questions asked? Are we using the appropriate controls, dosages, and characterization methods? And then what are the contributions of the experimental design to the response of the cells? And so with that, I would like to thank everybody for their time today. And if I can answer any questions, I would be happy to do so.
We did have some questions that came in, Dr. Reynolds. The first question, for therapeutics, how should cell culture models be modified for acceptance?
So I would say there's a couple of questions or a couple of considerations to think about there. I think one is to make sure that the cells have all of the nutrients required for growth. This tends to be a bigger problem with bacterial studies than it does for cells, but one of the concerns is, are the cells actually growing appropriately for the studies that are being conducted? The second piece is are the cells still functional even if they've actually grown appropriately? So there's many times that cells might have their normal appearance but then don't respond to biological stimuli that they would under normal conditions because a biochemical pathway has been knocked out. So when we think about creating cellular models, we have to make sure that the cells are going to be that they need to be functional after we do the treatment.
The second question that's come in is, how appropriate are in vitro cell models for predicting in vivo success?
So that is a great question. So, and that one is always, many conferences that I go to, that one comes up for debate all of the time. I would say that it's difficult to translate in vitro models to in vivo success. However, I will say that there are some important features that we can learn from in vitro models when they are designed appropriately that give us indications for in vivo performance or success. And I would say one of those is making sure, again, that the system is set up, and I love the idea how many groups are moving towards whole culture type of setup where you can actually then also consider the influence of having multiple types of cells present, which is in a closer mimic to what an in vivo environment would look like. So I think that's one important consideration. I also think that a lot of the in vitro models that are starting to come out now have a lot of potential for us to think about how they might affect performance in vivo, because people are considering some of the problems that I had pointed out today with current systems, and that is they're creating an environment that's not as simple and therefore more mimicking of what we might see in an in vivo environment.
Third question what are the important for scientific design for long-term versus short-term therapies involving cells?
So I think in terms of cellular experiments, one of the grand challenges is creating an experiment that actually looks at the endpoint of the target that the inventor or the investigator is looking at. So for blood contacting devices, a lot of times folks will look at 2-hour experiments and they'll say, well, that is indicative of the initial response. But these devices are implanted for 3 weeks, 2 weeks. And so how is that going to indicate to us longer-term performance? And again, for a lot of the cellular-based therapies, there's a growth phase that has to go through in order to create functioning cells. And then the question is going to be if those cells need to last or be reproduced or replaced or regenerated over time, how long does that need to happen? And can we create a paradigm that says if we grow for this amount of time and then see this behavior and we change that, will that be predictive of what might happen 3 months down the road? And so I think we need to start asking more questions related to the time domain of these experiments if we're really going to start thinking about longer-term therapies.
Well, thank you very much, Dr. Reynolds. We appreciate it. I'd like to thank everybody in attendance for the webinar. Dr. Reynolds has graciously provided her contact information on the screen for us. You can contact her regarding the presentation, or you can contact me at Bioquell. Also would like to thank our marketing director, Jennifer Portland, for putting the webinar together, and once again a big thank you to Dr. Reynolds for her time and efforts in participating in this format today.